Hi. So last time I gave you a brief introduction to the three major schools of taxonomy or systematics. That is, let's see, evolutionary taxonomy, phonetics, or what was originally called numerical taxonomy, and then phylogenetics. So what I want to do today is to go into a little bit more detail about uh, these schools, beginning with evolutionary taxonomy and phonetics. In the next video, well, actually, in two videos, we'll jump ahead and talk more about the techniques of phylogenetic systematics. So this is the reading I assigned for today. A 1986 annual review article by Robert Sokol on phonetic taxonomy, its theory, and methods. It's probably a little bit dense, and you may be wondering why, all you millennials, I would be assigning a paper for reading that is what, 14 and 21, 35 years old. The question would be, haven't we advanced since then? And on the one hand, I would assure you that yes, there have been lots of advances, especially since, as you probably know, in 1986, sequencing technology was only in its bare beginnings, and the explosion in the amount of sequence data and its how cheap it is and how ubiquitous it is these days has driven a lot of methodological advances, in addition to some philosophical advances along the way. But the reason I actually bothered to talk about phonetics is not from a purely historical point of view, but because you will still find methods of phonetic taxonomy uh, published in the modern literature. As a matter of fact, you will find phonetic techniques are some of the algorithms that the mega program has incorporated. Some of these are extremely common and very widely used. So it's important that for two things, that you be able to recognize what they are and have some idea what the underlying assumptions are, which will then be able to guide the way that you can interpret uh, the results from those methods, and to decide whether or not these are the methods that are appropriate for the question that you might be asking in your own work. So let's start with evolutionary taxonomy. Um, the first thing to note is that this idea, or this naming of this field of systematics as evolutionary taxonomy, is really sort of a backwards construction, a backward-looking construction, in that this is actually the oldest um, sort of philosophy of classification that goes back probably as far back as you want to go. And it was only with the rise of phonetics and phylogenetics and the criticism that the advocates of those methods made, that the people who advocated the traditional form of classification, the most prominent of whom was Ernst Mayr, but even within my lifetime, there were many people who were just um, absolutely opposed with all of their heart and soul about what phylogeneticists and phoneticists were proposing, and still advocated this. So it was only with this sort of challenge from an alternative way of doing things that Mayr and his associates started to try to articulate what they did, why they did it, and why their methods were, in their opinion, superior to those of other philosophies or schools of classification. So I guess the first thing we need to reiterate is, what are the goals of classification? And you'll find that the goals of classification are going to differ between these three schools of thought. So for an evolutionary taxonomist, well, for all of them, there are certain things that they hold in common. That the idea of a classification is an information storage and retrieval system, in a way. That the information, that is all the information about the characteristics that organisms have, their sequences, their behavior, their morphology, everything, that's information. And we want to be able to retrieve information from a classification to know that, for example, all tetrapods, at least ancestrally, had 4 legs. We want to be able to retrieve that information. We want to be able to predict the characteristics of unknown organisms. If we can place them in the classification, then we can figure out certain things about them. I don't have to look at every bird and study its biology in detail to come to the reasonable conclusion that the bird is going to lay eggs as a mode of reproduction. So that's what I mean by predictive aspects of a classification. So the real question is, if we do agree on those goals, at least those central goals, and there are some ancillary ones along the way, how can we best achieve those goals? And that is the crux of this intensive philosophical battle that took place, well, beginning probably as early as the 1950s, but running very hotly up until the 1980s, 1990s. It's only recently that it's sort of died down around the edges. And when I say this was an intense battle, I mean the people publishing papers were absolutely writing poisonous pen letters, if you will, against their opponents, and it was very emotional, very derogatory in some cases, but that did focus a lot of attention on the field and got people to think seriously about it. And it also brought a fair amount of disparagement from people working in other fields, saying, why the heck can't these systematists get their act together and work together in order to find the best methods in order to achieve their scientific goals. So evolutionary taxonomy. Ultimately, any of these taxonomic products is going to be based upon an intimate knowledge of the group in question. That is the characteristics of those groups. And so we run into some terminology that I want to probably not introduce to you, but at least be explicit about what I mean and what systematists mean when they talk about these. The first term is characters or taxonomic characters. A character is really any feature that varies among the organisms that you're studying, that you're trying to classify. It can be, I guess, the most important element of it, the most important limiting factor, if there is one, is that these characteristics must be heritable. That if we're talking about environmentally induced differences amongst organisms due to solar insolation, rainfall, any of these kinds of things, that at least that immediate manifestation of those environmental differences is not what we're interested in. I suppose that ultimately you could say that the reaction of the organism to these environmental influences, it may be in some ways driven by, constrained by, directed by their genetic makeup. And insofar as we can identify those factors, then those are fair game as characters for a classification. But if they're just, a tree grown in a dark forest, the leaves are going to be larger in area on that individual, whereas if it's grown in bright sunlight, leaves tend to be smaller. In that case, the immediate characteristic of the size, the area of that leaf surface, of the surface of an individual leaf, is not going to be something that we're immediately interested in. So a character is any feature, heritable feature, that varies among organisms. And the most accessible to these, of course, are things from the anatomy, the morphology, the structure of the organism you're studying. But, it could also be behavior, it could be physiological characteristics, could be sequence characteristics, anything, as long as it's heritable, is really fair game. And it needs to vary among the individuals in the group that you're studying. If they're all the same, then that's not a very interesting feature, at least for that particular group. If you extend your focus outwards, then maybe that will come into interest later on. So we have characters. And then what I'm going to call character states For example, what did I have before? I had an example of the roseate spoonbill, and that at least some of the feathers on a roseate spoonbill are pink in color. So we could say feather color is a character, and then have different states of that character: white feathers, pink feathers, black feathers, brown, blue, green, whatever. This is an example of what I would classify as a qualitative character type. So I'm going to that next bullet point here. Character states we usually think of in terms of qualitative characters. Let me just grab my pointer here. There we go. I had it at one point and I lost it. There we go. So in my simple example, the different colors, as long as I can immediately distinguish them. Of course, you can also, think of color. You could measure the spectrum, the reflectance pattern of the feathers, and actually quantify the wavelengths of light that are absorbed / reflected by a particular structure. And so perhaps the qualitative difference here between qualitative and quantitative characters is not absolute. Usually quantitative characters, we're talking about things that can be measured. And typical examples might be body length, tree height, the area of a leaf, those kinds of things. Those kinds of things that we can measure. And then we can measure the variation in a population to get average height, median height, variance in height or length or whatever in an organism. So qualitative is usually A or B, black or white, these sorts of categorical characteristics. Quantitative is something we can measure and put a number on. Meristic characters are actually, I suppose, a subset of quantitative characters. Meristic characters are things that you can count. So if you study fish, one of the common characteristics that people use are the number of rays in a fin, the number of cartilaginous or bony support rods that are in a bony fish. So meristic characters are things you can count: the number of wings, the number of legs, the number of antennal subsegments, and things like that. There's not really, these all can, I think you can see that these all do blend in to one another at the edges, but it's often useful to think about them differently. And when we get into actually developing algorithms for our classificatory techniques, these different kinds of characters have different sorts of characteristics that we need to model into our algorithm. Now, another aspect of evolutionary taxonomy, which turned out to be one of the huge bugaboos and just taboos, if you will, when the phoneticists came to the fore, was the idea of character weighting. So, in this case, evolutionary taxonomists said, well, we can, if a character is anything that varies amongst organisms, some of these variations might be very trivial. Does the antenna of an ichneumon, one of these big parasitic wasps, have 25 segments or 26 segments? That's different. Is it significantly different? Is it important how different than they are? Those differences are in comparison to, say, how many wings does it have? Does it have 2 wings or 4 wings? You know from your basic insect taxonomy that if insect or no wings, right? If an insect has no wings, then that either implies that they've lost wings in the course of evolution, things like, what do we have? Fleas and lice and things like that. And because we look at their closest relatives and see that they have wings, we interpret that they lost their wings as a consequence of adopting this parasitic lifestyle. Do they have 2 wings or 4 wings? The most common group of, for FOUR, of insects that have 2 wings are of course the Diptera, the true flies, but then there are other groups that have only 2 functional wings, Strepsiptera, for example, male scales, for example, and things like this. And so this is that changes in character and the number of wings that organisms have is in a way much more fundamental, much more important in indicating something about their evolutionary history than whether they have 25 or 26 subsegments on the antennal flagellum. So an evolutionary taxonomist would say that we should give more importance in our classificatory decisions to those characters that are more important, that have more weight. And this idea of weighting, of giving more importance a priori to one set of characters versus another is something they held very near and dear to their heart, and which some other groups of taxonomists criticized very strenuously. And the basic idea behind the criticism was that if I construct a classification and then look at the distribution of character states, then I can see which characters in that classification are important in determining its structure. And so then I can say a posteriori, after the fact, that those characters are important and should be weighted, should be given extra emphasis. But before I start, how do I know this? If I start out by saying that the number of wings is important before I've studied anything, then I am biasing myself in the process of coming up with a classification. And that would be, in many circles, considered to be antithetical to the scientific method, that the person doing the work should try to eliminate as much as they can their inherent biases. The evolutionary taxonomic point of view that some things are more important than others, and we can know that in advance, was just antithetical to some other people. So I think the basic core of what we describe as evolutionary taxonomy, and I'm sure I mentioned this in the last video, is that in their classification, they think a classification should represent the two different fundamental aspects of the evolutionary history of an organism. That is the to put it in modern parlance, the relative recency of common ancestry, the branching pattern on the evolutionary tree, and it should represent how much change has occurred along each of these branches. That illustration I have there on the right is Darwin's original, and I'm not sure, perhaps only phylogenetic tree that he ever published. This came from the Origin of Species in 1859. So evolutionary taxonomy seeks to do both of these things. They believed that both aspects of evolution are very important, and both should be reflected in the classification in order to be able for that classification to serve its general purposes as an information retrieval, storage and retrieval system. What we today would call phylogenetic trees or tree-like structures actually predate Darwin by quite a bit. People were drawing these hierarchical structures to represent their classifications, and classifications typically took on a hierarchical structure long before Darwin. But after Darwin, the idea of evolution by natural selection, this gradual change and accumulation in ancestral descendant relationships, was basically the underlying justification for the structure that classifications took. So everybody wanted to represent common ancestry in their classifications. They wanted to be able to say things like, well, vertebrates are classified together, and one of the reasons is that because they all evolved from a single common ancestor and then diverged in their various directions. Simultaneously, they also wanted to represent in their formal classification, and maybe I need to step back here half a second and reiterate that when I talk about classification here, I'm talking about this formal Linnaean classification, the putting of organisms into ranks, into species, genera, families, orders, classes, and things like that. So it's quite clear in a number of instances that evolutionary change does not proceed uniformly in the two species that might arise from a single common ancestor. That is, one species might change very little and basically stay the same, while the other changed a tremendous amount and became very different. The classic example of this is the classification of vertebrates into classes, and specifically the idea of the classification of birds. It has long been accepted that birds and reptiles, as we currently recognize them, evolved from a common ancestor. And we'll get into the question of monophyly when we get into phylogenetics. I know you guys all have the terms monophyly and monophyletic at the tip of your tongue that you've been taught them for years. But let's just hold that in abeyance for the time being. My point here is that everybody recognized that birds evolved from a reptilian sort of ancestor that is exemplified in numerous characteristics, in the scales that they have, lots of anatomical characteristics that birds have. But the idea was that they had changed so substantially in their evolutionary history from what that original reptilian ancestor was in comparison to other things that we now classify as, or classified as reptiles. I need to use the past tense there. That they deserve to be recognized as a distinctive branch on the phylogenetic tree. And I don't think there would be anyone who would argue that there had been much more anagenetic change along the lineage that leads to modern birds than there had been in the other lineages that were also at one point classified as reptiles. That's not under contention. What's under contention is whether or not, by virtue of that tremendous evolutionary change that had taken place in that lineage, that we should recognize birds as a higher level taxon, on the same level that we recognize mammals and reptiles and fishes and things like that. So those are the two aspects that evolutionary taxonomists want to incorporate in their classification. Both the idea of common ancestry, so we want groups that evolve from a common ancestor, but we also want to be able to reflect the amount of evolutionary change that has taken place within a given lineage. A couple of corollary ideas that go along with this. One is the idea of gaps, kind of a measure of the distinctiveness of a taxon that you recognize in your classification. Sorry that my picture is overriding the words here. You'll be able to see this on the PDF of these slides. So gaps are a measure of this evolutionary distinctiveness of anagenetic change, and it's a very basic idea. What is the point of classifying two sets of species into two different groups if you can't distinguish those groups? If the groups are indistinguishable, then what purpose does that serve? If they're indistinguishable, they should just be grouped together. So that's the basic idea, and this is kind of a working rule of thumb for taxonomists from the evolutionary taxonomic school, and I think it still has some influence today. If you're classifying a group of closely related organisms, then generally the idea is that the size of the taxon that you have should be inversely proportional to its distinctiveness. In other words, if you're just dealing with a few species and you're trying to figure out how these should be best classified into genera, if you're only dealing with a few, 5 species, then they better be pretty distinct in order to put them into different genera. Whereas if you're dealing with a group of 800 species, like the carpenter ants and their relatives, then in order for this classification system to be functional, for it to actually be practically functional, then you're willing to tolerate that the genera in campanotines and their relatives could be very similar, only distinguished by relatively small gaps. So large, diverse taxa, more willing to tolerate smaller differences among genera, whereas very depauperate in terms of number of species. If you're dealing with groups that are very depauperate in terms of number of species, then really to justify putting them into different genera, they've got to be pretty distinctive. We kind of see this today. I just read this article recently, or at least an article about an article in the New York Times about dire wolves. And so let's see, dire wolves, prehistoric canids in North America. Apparently there are lots and lots of fossils of these things, especially from the La Brea tar pits in Southern California. But they've only recently been able to get enough DNA. Apparently tar is not good for DNA. They've only been recently able to get enough DNA in order to get a reasonable estimate of the genome of these creatures. And whereas they used to be thought to be very closely related to wolves and were placed in the same genus as they are, now the researchers found that their genome is so very distinctive from wolves that they are proposing that they be placed in a separate genus from them. At one scale, well, in one way of thinking, this here we go back to the old evolutionary taxonomic school of taxonomy, because this is basically saying we should recognize these animals as a different genus, not because they don't share a common ancestor with the various species of wolves in the world. They do, and that's recognized. But that common ancestor, well, one way to think of it is that common ancestor is more remote than we thought. That's okay. But really the underlying justification was that these things, at least in their genome, not so much in their skeleton, but at least in their genome, are so distinctive from wolves that they deserve, I say that word with a lot of negative attitude in my voice, they deserve to be put into separate genera. Frankly, I don't agree with that, but I'm not going to get into a shouting match with a bunch of vertebrate paleontologists. One of the problems with evolutionary taxonomic classifications is that we have these two competing criteria for how to group organisms into higher taxa. Common ancestry and evolutionary distinctiveness, the amount of anagenetic change. And these things are, in some senses, well, there's also the weighting issue. And these things, you run the danger of the decisions being made about how to classify organisms is highly susceptible to what I'm calling an appeal to authority. That is, there may be very few people in the world who know the groups in question to be able to specifically articulate what they have in common, what's different about them. And these authorities tend to get more credit for quote unquote knowing what the right classification is than they might deserve. Then they could reasonably justify in an objective manner. Also, I kind of stepped in my line here. One of the other problems with evolutionary taxonomy is that if you're trying to include two aspects of evolutionary change, cladogenetic change and anagenetic change, in the same common hierarchical structure, in the same classification, then somebody looking at that classification without the underlying data has no way of knowing if a taxon is being recognized because here are all the descendants, sorry, of a common ancestor, or these guys are put in a higher classification because they're very different. They share the same common ancestor with these over here that are placed in a different group. But because they're so different, I'm classifying them differently. And so we can't know how, if we simply look at the words that make up a classification, the names of the organism, we don't know if a group is being recognized on the basis of its unity, on the basis of common ancestry, or because it is so different than other organisms. Which leads us to the 1950s and to the advent of probably the first, at least in the Western scientific world, severe criticism of, or ineffective criticism of the traditional methods of classification. And that is the field of systematics that we call phonetics. As I told you last time, in the early days, this field really got prominent status because of a book that was published by the two gentlemen there on the left of this image. It's Robert Sokol on the left and P.H.A. Sneath in the middle. And the book was called Numerical Taxonomy. So phonetics, numerical taxonomy, in many cases ended up being synonymous, although as we go forward a couple of decades, phylogeneticists were also using computer algorithms in order to handle and analyze their data. So in a sense, they were also doing numerical taxonomists. Maybe you could even argue that some of the evolutionary taxonomists, insofar as they were quantifying their information and analyzing them using computers, could be called numerical taxonomists. But originally, that term was pretty much limited to what I'm going to be referring to here on as phonetics. Now, I think I made a mistake last time when I talked about Sokol and Sneath as two of the founding most important members of this field of systematics, and I said that Sneath was from Stony Brook. I was confusing Sneath with another prominent co-author of Robert Sokol, and that other person was James Rolfe. So Sokol and Rolfe published a very influential book on statistical techniques. Sneath was a bacteriologist. He was British. So the third guy I've got there actually, I think, went a long way to giving Sokol and Sneath the credibility within the wider biological community for what they were proposing as a revolutionary approach to classification. So Sokol was basically a mathematician. He did some interesting work looking at variation in a gall aphid, of all things. So there is a gall aphid of the genus Pemphigus. If you find like a cottonwood tree, you'll very often find you got the leaf, it's kind of a heart-shaped leaf, and then the petiole coming out. There'll be a little spherical gall at the base where the petiole meets the leaf. Living within that gall is a tiny little colony of aphids, and so Sokol got into studying the variation and evolution, I shouldn't say evolution, the variation and population structure of those aphids. So he did have an entomological connection there. But the real person who grounded the work that Sokol and Sneath proposed in issues in the real biological world was the gentleman on the right, Charles D. Michener. Michener was a grand old man of systematics. Sokol and Michener at the time were at the University of Kansas, and they collaborated on a number of papers. So Sokol might develop techniques, and then they would be applied to Michener's particular group of interest, which I know we have several people in the class who are interested in bees. I'm sure you have run across the name of Charles D. Michener. He lived to a ripe old age. He was attending international congresses well into his 90s. Everything. One of the members of my graduate committee was a student of Michener's, And then I had a colleague here at Ohio State for many years who was also a student of Michener's. They, to a man, to a person, because he had many female graduate students as well, to a person, they all describe him as one of the most kind persons they'd ever met. Very soft-spoken, very gentle. It was with some trepidation that I had written up a manuscript in an area that I had, very little prior experience, and Michener had published a little bit about this, and I sent him a copy of this and asked him for comments, and he was generous enough to both say kind things and to actually do the review for me in pre-publication stage. So I was very, very grateful for that. Michener, tremendous bee systematist, also did a whole lot of work on bee biology, nesting behavior, interested in the evolution of sociality amongst bees, going from semi-social, social to eusocial bees, and solitary bees, of course. So these were three of the big names in phonetics. There are quite a number more, as you can imagine, but these are the names that come to my mind when I think of the development of phonetics as a challenge to, as a separate way of approaching the problems of classification. And make no mistake about it, there were real problems in classifications. So, underlying principles of phonetics. Ultimately, we want to get to the same point. We want to have a classification that is an efficient storage mechanism for information, and by which we can get information out. And it's predictable. We can predict things with some accuracy about organisms because if we know their place in a classification. However, in contrast to the evolutionary taxonomists, as its name implies, the phoneticists believe that the evolutionary history of organisms themselves is not directly observable. You can observe they did believe in evolution, don't get me wrong there, but the process by which organisms evolved, the steps they went through, that's not directly observable. We can observe the consequences, the later impacts of that in the features that organisms have today, but we can't see the process directly. And it's not measurable, that process of evolution. The consequences are, but not the process. And so they harken back to, one of the major characteristics of what science is in the first place, in that it's empirical, that it's based upon our ability to observe, to measure, to quantify to some extent the natural world, and that what I'm seeing, if I'm specific enough, you can also see and measure. If I say the temperature is, 30 degrees centigrade, you can get your own thermometer and measure the temperature, and hopefully it'll be close to my 30 degrees centigrade, at least insofar as our thermometers are compatible. That there is something objectively in the real world that we can observe. And when I say observe, I mean that in all of its dimensions, including quantifying what I'm observing. And we see this inevitable trend in science toward quantification in order to try to eliminate the interpretation that the human mind inevitably imposes upon its observation. So if we can get, air quotes here, objective quantitative measurements, things that's more reliable than a single person's evaluation of it's rather hot today. So we're not going to explicitly include evolution in the formation of our classification. Evolution's out there, but we can't know what that was, so we can't include it. Secondly, if we're going to recognize groups of species as separate taxa, we should base that grouping on an assessment of what they refer to as the overall similarity of the organisms that you're classifying together. By overall similarity, I mean the totality of all the characteristics that organisms share in common. With the idea that if you're grouping species together within a single higher taxon, then one would desire that the species that are included in that taxon are all more similar to one another than each one of those is to a species that's outside of that group. So we have some homogeneity, relative homogeneity in comparison to outside groups. Which begs the question of what is similarity? We need a large number. If we want to measure overall similarity, then almost by definition, we have to be looking at a number of different aspects, a number of different dimensions of the characteristics that organisms share. These characteristics should be empirically observable, and the number of characteristics, I mean, in an ideal world, which everyone admitted does not exist, but in an ideal world, each characteristic would be independent of all other characteristics. But we know characteristics are related to one another. So to overcome that correlation amongst characters that are all kind of pointing at the same thing, perhaps, we should be looking at a large number of characters so that we're getting a comprehensive sampling of all the characteristics of these organisms, so we can then talk about overall similarity rather than just similarity in wing structure or similarity in, I don't know what, mouth part structure. We want all characteristics because we want our classifications to be general, not to be a special classification of the different types of wings in insects, but all of the characteristics of insects. And it's probably occurred to you that going along with that, if we're coming into this exercise, we don't want to be imposing our subjective impressions of which characters are more important than which other characters. In other words, there should be no character weighting. I think I left that word off there. No character, a priori, is more important than any other. So characters should not be weighted beforehand, before you produce the classification. If you're then going to translate a classification into an identification scheme, then you would concentrate on the characters that are going to enable a person to classify them most effectively. So a posteriori, sure, you can have weighting, but not when you go in initially. All characters are potentially of equal value, therefore they should be, another way of saying no weighting is to say that they are equally weighted. So, back to similarity. How do we quantify similarity? How do we measure it? If we're going to be looking at large numbers of characters, and our objective is to quantify how similar they are to one another, we're going to get into computations, and because of the computations necessary in the techniques of phonetics, I suppose you could do them by hand, but unless you're like a green-eyed shade Victorian bookkeeper who can do all the math in their head, computers are going to be an essential tool in analyzing these data. And so it's no coincidence that phonetics started to arise and gain traction in the late 50s, early 60s, when mainframe computers started to become available. So again, that's the reason they got this moniker. Well, actually, Sneed and Sokol entitled their book Numerical Taxonomy, because they were analyzing things quantitatively. So let's take a look a little bit more closely about what we could mean by similarity. So I want to walk through the steps of constructing a very simple similarity index, such that I can, in a single number, express how similar one species is to another species. So that's all I mean by an index, a measure of the similarity here. So let's assume that we have a set of characters, and these characters are qualitative characters, and they're binary. That is, there's only one or two states. There are only two states, which we can express them any way you want. You could call them A and B, black and white, left and right, I'm going to call them 0 and 1. But the coding, how you express those two states, is absolutely trivial. But because we're going to be using math in order to express this as a number, obviously it makes more sense to use numbers in order to express what these characters are. So we have a set of characters, and they're all binary in nature. So here, whoops, forget this slide, forgot to delete that one. Here I've taken two species, species A and species B, and there's that term that I mentioned in the write-up for the first lab there, OTU. All OTU stands for is Operational Taxonomic Unit. That means in my analysis, I have a number of different groups that go together. In this case, I've called them species, but they could be genera, they could be families, it could be subfamilies, it could be a mixture of any of all of those in terms of taxonomic rank. They may have no taxonomic rank. It must be a collection of individuals. Operational taxonomic unit is used as sort of a blanket term so that you don't get involved in worrying about formal rank in the Linnaean hierarchy too soon anyway. So for OTUs, in this case, in this table here, I have just a pair of species, pair of OTUs. And then I've listed 10 characters as the columns in here. And basically, I went to the rand function in Excel and generated some random, or at least Excel's approximation to random numbers for the binary characters that these two species will share. So we see for character 1, species A has state 0, species B has state 0. Character 2, species A has state 1, but this time species B has state 0, etc., etc. So the question is, how can we express quantitatively in a single number how similar species A and B are? If we want to express it in multiple numbers, we just have the two vectors here of their character states. But I want to express it in a single number. I want to encapsulate all of the all the shared characters and all the dissimilar characters between these two. So in this simple-minded example, we could simply look at, well, how many character states, or what percentage of the character states of these two species have in common. And we can see in this case that there are 4 character states. Let's see. They both have character 1 with state 0, character 5 with state 0. Don't interpret anything in the zeros and ones here in terms of any of these ideas about ancestral, derived, primitive, etc. We'll get to that soon enough. Right now, we should be totally agnostic about what these numbers mean. The question is, do they match or do they not? So character 1 they match, character 5 they match. Did I miss one? Character 10 they match. Character 8. So there are 4 character states in which they match. So one way of expressing their similarities is just to say in all of their characters, they match 40% of their character states. So you could say that they are 40% similar, or express that as an index, the similarity between species A and B equals 0.4. Also point out that this is, commutative. Species A is 0.4 for 40% similar to species B, and species B is 40% similar to species A. You can see they're going to go both ways. So let's make this a little bit more complicated, not much, because I still have random data. But in this case, I'm still dealing with binary data. I'm not going to get into it, but you can, if you are inclined that way, generate your own similarity matrix where you don't have just binary data, but you have what we call multi-state characters. So instead of just 0 and 1 being the potential character states, you might have 0, 1, and 2, or 0 through 10 as the different character states. And so again, with the Excel random number generator, I've put together some data here for 5 species. Species A, B, C, D, and E. Those are my 5 OTUs. 10 characters. Now, in the same way that we express the similarity between just that pair of species in the last slide, we can ask, well, how similar are species A and B in this new slide? How similar are species A and C, A and D, A and E, B and C, blah, 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 up until we've made every possible pairwise comparison amongst these 5 species. So we'll probably want to express our similarity index a slightly more formal way. Again, we're still basically dealing with binary characters here. But if we let x represent a character and x sub I be the character state for character x in species I, and x sub j be the value of the character state for character x for species j, and let n be the number of characters. So in our previous example, n is going to be 10 because I have characters C1 through C10 there. Then if we want to do that same calculation we did in that original 2-row table for every possible pair. So we want to, one way to do it is to, since now we basically want to generate an algorithm to do this for us, because you'll probably find that I've made some kind of arithmetical error in the example I'm going to present, and it's very easy to do, and so it's much easier to do consistently and correctly if you have a computer do it for you. But basically we have 2 characters for we have 2 species, and we look at each individual character. So for character I, for species I, we look at its value. Species J, we look at its value, and we compare them. If they both have 0, then the difference between those numbers is 0. If they both have character state 1, then the difference between those 2 numbers is 0. If species I has 1 and species J has 0, then the difference between them is 1. Species I has 0 and species J has 1, then the difference between them is -1. So you see that if they're the same, then the difference is going to be 0. If they're different, because it's a binary character, it's going to be either +1 or -1. But the sign there is really meaningless. What we're focusing on is that they're different in that character. So let's get rid of that sign when we compare the values for that character for those 2 species. And you can do it one of 2 ways. You can just take it as the absolute value of the character difference, or, and this is where it becomes valuable for more generalized forms of this simple matching coefficient, is to take the difference, square it, and then immediately take the square root of that, the positive square root of that. So, and then you do that for every character for those 2 species. Get the average distance then, average distance difference. I probably should have said difference here first. Get the average difference in character states between species I and species J by taking the sum of the differences and dividing it by the total number of characters. I'm going to bring it up here a little in a second probably, but since I've already said it, similarity and distance are kind of the same things. You know, we can say 2 things are similar or 2 things are dissimilar. They're like different sides of the same coin. And when you think about similarities and differences in a graphical context, you can think of it as the distance between those 2 species on the axes that are the characters between them. So distance, if 2 things are close, it means they're similar. If they're far from one another, it means they're highly dissimilar. And both similarity, dissimilarity on the one hand, and close and distant on the other are really 2 sides of the same coin that you can interconvert between them very easily. So, so far the index we've produced, which is to look at the difference between the character states in 2 species across all characters add them all up, divide by the total number of characters. That's really a measure of the mismatches. That is, it's a measure of the dissimilarity. And we could go with that, but it's really easier for me to talk about their similarity. So in order to convert, because we've got this normalized so that its value scales from 0 to 1, let's just divide it, let's just subtract 1, the index we've calculated, from 1, so that if there is no distance, no difference in the character states between two organisms, then this component over here, the sum over all characters of the square root of the square of the difference in each and every character divided by the total number of characters. This is going to be 0 if they're exactly the same. And so the similarity is going to be 1, 100%. If they're different in every character, because our characters are binary, then this is going to be equal to 1. Subtract that from 1, you get 0. The similarity is 0. They are entirely dissimilar. So I knew this was coming up real early, but I guess I spilled the beans, stepped on my punchline a little bit earlier. So now we want to use this little simple matching coefficient to measure across all species pairs. And to generalize this, we would incorporate yet another subscript here. But let's walk through this. So I am converting here my original matrix, which is a taxon by character matrix. So it's got each taxon as a row, each character as a column, and the value of that character in each cell for each species. What I'm doing here is simply counting up the number of differences between the two. This is the xi minus xj matrix. So now basically I'm comparing one species with another species. So I have S1 through S5 and S1 through S5 as both the columns and the rows. So obviously S1 perfectly matches itself. There are no differences when you compare S1 with S1. So that's 0. And similarly, you see all along this diagonal where you're comparing an OTU with itself, there's going to be no differences. And so this is 0. And because I mentioned before that I get it right, that the differences are commutative, that it doesn't matter which direction you look at them, we see that this matrix is actually a mirror reflection of itself along that diagonal. So for all the rest of them, I'm just giving you 1/2 of the matrix, in this case the part above the diagonal. So here I've simply gotten rid of the mirror copy down below, and here I've divided by the total number of characters, because the example I've concocted had 10 characters. It's just a very simple, it looks almost exactly the same, except I put a decimal in the numbers here. And from here, this is mismatches. Now I want to convert that into matches. So from dissimilarity to similarity, basically subtracting these values from 1. So now my diagonal is now converted. S1 is perfectly 100% similar to S1, S2, S2, etc. So that's what this is. This is basically a distance matrix here. Or, if you care to think of it that way, a similarity matrix. Just as a sidelight, there are many different measures of similarity that one could use. More than a handful. You could have correlation coefficient here, depending upon the nature of your data. There are many, many different ways of expressing similarity, and that's why I chose this just kind of brain dead, very simple matching coefficient and as an expression of similarity here, because we're going to walk through, calculate it, and understand the logic behind it, and by extrapolation, understand what's going on in other when we use other metrics of similarity. So I now have a similarity matrix showing how similar each species is to every other species. What are we going to do with that? Basically, we want to convert, in a sense. We could stop. we could stop right there in that similarity matrix. But it's a little bit difficult to, for me, it's more than a little bit difficult to translate that similarity matrix, especially if you're talking, you might have dozens, hundreds of species, to translate all of those numbers into some sort of coherent representation of the overall similarity between all of those species pairs in one swell FOUP. So we want to apply a clustering algorithm in order to do this. And again, I'm going to give you probably one of the most basic clustering algorithms and talk a little bit later on about other options for this agglomerative, these agglomerative techniques, a way to pull these things together. So here in this illustration here, that's the same similarity matrix that I ended up with last time. And at this stage, we want to look at the values in the similarity matrix and find the pair of species, to find the pair of OTUs that are most similar to one another. So we peruse through this, and you can obviously see that I found the answer is 0.7. S2 and S4 share 70% of their character states. Just as a sidelight, because these numbers are randomly generated, the range here is not going to be very high. You'll probably find higher ranges when you look at real organisms. But I find that S2 and S4 are the most similar pair. So I'm going to group them together. I'm going to pull them out and group them together, and that's my first grouping. Now that I've grouped these together, I need to find S2 and S4. My question becomes, how close is S1? How similar is S1 to the group of S2 and S4? How close is S3 to the group of S2 and S4? S5 to the group S2 and S4? And to do that, basically, I'm going to simply take the arithmetic average of these original similarities. So for example, how similar is S1, species 1, to the group S2, S4? Well, let's see. S1 is the similarity index value is 0.4 for S2, and for S4 it's 0.45. Take the arithmetic average of that, and that would be 0.45. Do that over all combinations. Now I had to go back and review my math because I found an arithmetical error, and in doing so I discovered that I misspoke in that last slide, is that the similarity was not 0.45 but 0.5, I believe, to end up with an average of 0.45. Anyway, here we are at the next point where I've plugged those numbers in to the similarity matrix. There we go. So I had 0.45. This number is from the original, this number is from the original, this number is from the original, but these are recalculated for this group, just being the mean of the similarity of each of the now 3 components. Again, we look through the matrix, you find the highest similarity value. In this case, it's going to be between S1 and S5 at the 60% level. So we group S1 and S5 together. The next step, just more of the same. Now we're going to recalculate similarities. I'm going to find the similarity of the group S2 and S4 to the group S1 and S5, and then the similarity of group S3 to S1 and S5. We already have S3 with S2 and S4 here, and we generate a new matrix. Find the highest value, 0.45. That groups us S2 and S4 and S1 and S5. So I'll now connect these 2 branches, leaving S3. So we know that's the last branch coming off. And if you do the calculation, the level of its similarity is, I believe, 0.375. Now, we can all put all this together. And now, because actually I had values, right? That initial grouping was 70% levels. So now I've got a scale bar down here that looks like it's that PowerPoint has shifted it just a tad, but that's supposed to be 70%, 60%. What did we have? 0.45, so 45%, and then 0.375. Yeah, it's shifted everything over just a touch here. But that's the basic topology of the relationships here, and it's scaled, so I can tell what the values are. So that method that I just exemplified is something that you will find in widely used. You'll find it in your mega implementation. This is called UPGMA, which stands for Unweighted Pair Group Method using an arithmetic means. So you can see we're doing everything by pairs, and we're calculating our distances on the basis of the arithmetic means of all the members of those groups. This is just one clustering technique among a wide variety of clustering techniques. Another one that you'll see extremely commonly is something called a neighbor joining tree, which is basically the same idea. You have a matrix of distances. You are going to group things in a pairwise method, find the two that are closest, put them together, recalculate all the distances, find the two that are closest together, recalculate, put them together and recalculate, and go through this until you have no more OTUs left, that everybody is joined into this tree-like structure. As I say, neighbor joining is very commonly used. One of the reasons that it's commonly used is that it is numerically quite efficient, as opposed to some of the methods that we're going to be seeing once we get into phylogenetic techniques. Now, from a phoneticist point of view, the positive points of this method of coming up with a classification is that the methods are explicit. UPGMA, the steps are very explicitly known and articulated. The similarity matrix, you know how you constructed it, what the algorithm was for calculating similarity for that matrix. And so I can give the same set of data to somebody who knows nothing about the group, tell them, okay, you're going to use a simple matching coefficient on these binary data and construct a UPGMA tree. They will come up with exactly the same results, minus my arithmetic errors. So it's unbiased by the researcher. These are all very positive attributes from the point of phoneticists that address the shortcomings of traditional / evolutionary taxonomy. Gets the authority out of it. It gets the subjectivity out of it. And one researcher will come up with exactly the same results as another. Now, that tree, of course, I should be careful that hierarchical grouping of OTUs, tree has lots of connotations, which we'll get into, is itself, in some senses, it's a classification, but in terms of translating that into the Linnaean hierarchy, where do we draw the line for which species should be grouped together as a genus, which species should be grouped together as a family? Well, the phoneticist approach was to say, well, we could take specific values of similarity or specific values of distance and say that if you're this much close, then you belong in the same genus. If you're that much close, you belong in different families. So for example, if we say that species that are 70% or more similar should be placed, I only looked up the more sign, in the same genus, and anything 50% or more similar should be in the same family, then we can take our little exercise we just completed, and here's the 70% marker, here's the 50% marker. So we would say that species 2 and species 4 should be in the same genus because they're more than 70% similar. Species 1, species 5, and species 3 should each be in separate genera. But species 2 through species 5 should all be, let me make sure I got this right. No, species 2 and species 4 will be in the same family. Species 1 and species 5 will be in different genera, but the same family because they're more closely related than that 50% criterion. Species 3 is still down here, 0.375. Ignore that line. So it's in a separate family. So we'd have 3 families and 1, 2, 3 genera. Is that right? No, 1, 2, 3, 4 genera, and of course 5 species in this example. So we would have explicit values. We would know exactly where to draw the line of where where we should group the species into genera or in any higher taxon. So having said all that, and I've tried to say this, emphasizing the positive aspects of phonetics, I guess it's time to give you some cautionary notes. So we are trying to reflect overall similarities as expressed in a distance metric. I've given you a simple metric, that simple matching coefficient, very straightforward, that you could extrapolate to use with non-binary characters, and perhaps if you're clever enough, you have quantitative characters and heuristic characters, things like that. There are there's more distance metrics than you can shake a stick at, and the question is, which distance metric should I use? And to that, there is not a straightforward answer. Similarly, we did UP and UPGMA. You could do an unweighted pair group method with geometric means. You could do a neighbor joining tree. And there are a whole bevy of clustering techniques. Which one is best for you? I don't know of any objective criterion by which to select among the possible clustering algorithms. So then thirdly, I mentioned this idea about a tree. The mere fact that we use the word tree implies that these things are interconnected in some way and one giving rise to the other, and we often think of phylogenetic trees. Even though that UPGMA hierarchy that I showed you in the previous slide looks like a tree, it is not a phylogenetic tree. It is only reflecting overall similarity. It is not reflecting relatedness. Well, I should say, it is not necessarily reflecting relatedness. Insofar as overall similarity is mirrors the phylogenetic relationship. I've got to be careful here. Insofar as the overall similarity reflects the evolutionary history of the organisms, and to some extent you would expect it to, but not necessarily. Insofar as it does, though, then yes, you could interpret this as indicating the evolutionary history of the organisms, but it's not necessarily so. That's only one possible outcome of many. So that is not a phylogenetic tree, and it is not at all uncommon for you to see trees, especially people are constructing gene trees and the like, for them to use a technique like neighbor joining, which is a phonetic technique, and interpret that as a phylogenetic tree. That is absolutely wrong. And then finally, I've translated that similarity matrix into a hierarchy, which is basically a one-dimensional hierarchy. It looks like it's in 2 dimensions, but it's actually just measuring 1 dimension, the overall similarity. Why should I restrict myself to 2 dimensions? Why not, or 1 dimension? Why not 2? Why not 3? Why not as many dimensions as there are characters? And there are techniques to do this that you may be familiar with. These are techniques that you commonly see in morphometrics, or you see in ecology, multivariate techniques. This particular example is a principal components analysis, in which the idea is that you take a large number of non-independent characters, or I shouldn't even say characters, non-independent measures, and through the magic of algebra, you're able to convert them into a linear combination of variables that are, each variable is statistically independent of the other, so that we can now represent them legitimately as orthogonal axes here in 2 dimensions. And the other neat thing about principal components analysis is that the first principal component here labeled as component 1, accounts for the largest proportion of the variance in the original data set, in this case 42.89%. The second principal component accounts for the next largest component of the variance. What is it? 29.93%. Here's component 3 versus component 2 in this same analysis. These are a bunch of tree shrews, I believe, where the third principal component accounts for 11.06. What they've done to illustrate, because it's hard to do in a two-dimensional page, they've taken data for 3 dimensions and expressed it as dimension 1 versus dimension 2, dimension 2 versus dimension 3. You can imagine a 3-dimensional graph with like little balloons popping all over to show how things are grouping together. But there's no limit to this. Why we are not restricted to 2 dimensions? Why is our classification hierarchical in the first place? We'll talk about that a little bit later. But at least from a phonetic point of view, when we're measuring overall similarity, that single dimension of a hierarchy produced by a neighbor joining tree may not adequately represent, may actually distort the original similarity matrix in order to cram all of the information in that n by n, where n is the number of species, similarity or distance matrix. It may that one-dimensional hierarchy may actually distort those numbers, and so actually be misleading, and that this might be a more straightforward and objective way to represent the same data. So the basic problem here with phonetics is that there is a multiplicity of techniques that you can apply to the data without any objective criterion for selecting amongst them, and so that becomes a problem. So just to summarize here, evolutionary taxonomy, the principle is that the classification in our Linnaean classification should incorporate information from both of the 2 dimensions of evolutionary change into the branching on the phylogenetic tree, a real tree in this case, and the amount of change that takes place along those branches. The problems with this become, well, not only is it sometimes difficult to distinguish where the subjectivity of the individual researcher, but how do we tease apart these 2 components? How do we know when it's a taxon is based upon anagenetic change and when it's based upon its evolutionary history? It's not clear. It can't be clear because they have been merged into one. And then there are no explicit criteria by which to say, well, this grouping should be recognized as a genus. This grouping should be recognized as a family. There are no explicit criteria. And so these are the kind of criticisms of these problems that phonetics brought to the fore. Phonetics as a way of classifying, basic principles are that the groupings that you make should be based on the overall similarity of the organisms. No weighting. A priori, no character is more important than another. We should use as many characters as we possibly can. Conceptually all, although I think there's probably no practical limit to the number of ways in which characters can be broken down and interpreted and coded. They have explicit, repeatable methods. Let me step back there about that many characters business. You often find classifications, especially in the older literature, where tremendous emphasis is placed upon one character, one sort of character. For example, Fabricius, one of the classical, very important, very early insect taxonomists, put a lot of emphasis on insect mouth parts, where they had chewing mouth parts, piercing, sucking mouth parts, etc. A phoneticist would say, well, that's fine and dandy, but you're ignoring all the information from the rest of the body. So by using many characters, we're getting a measure of the overall similarity, not just a classification of mouth parts that we then plug all our species into. To go back, another advantage of the phonetic methods is that they are explicit, they are repeatable, they are not dependent upon authority. 2 people using the same methods, using the same data, we'll come up with the same answer, which I guess we would hope to always use. The problems are which of the literally hundreds of methods that are out there for calculating distance, for turning these into hierarchies, for representing these data, which ones are best? And there's no way that what might be best in one set of circumstances is not necessarily the best in another. And how do you deal when you take the same data, use two different techniques, and you come up with different answers? Which one is quote unquote right, or which one is best? Not clear. And then finally, that principle that the evolutionary history of an organism is not directly observable, and therefore we should eliminate it from our classification. The problem here is that evolution is exactly what most of us are interested in, and to just divorce our classifications and our organizational principles from the thing that we're trying to understand seems counterproductive at best. So not surprisingly, there were many adherents of phonetics, and even more people recognized the validity of the criticisms of traditional and evolutionary taxonomy. But the phonetic techniques left a lot to be desired in terms of coming up with a single general best classification for organisms. So I'm going to leave it there for today, and render this video and get it uploaded for you for tomorrow. And then I'll see you next time. Probably the next video is going to be not posted tomorrow, but on Wednesday. So until then, take care. See you soon.